Hello students, I am Dr. Kausalya, Professor of Food Science and Nutrition from the prestigious Avinashalingam Institute for Home Science and Higher Education for Women. The topic that we are going to see today is concepts of nutrigenomics and its applications. Hope you are able to recall the lessons on the basic gene structure, chromosome structure, and also the lessons on recombinant DNA technology. How do we introduce the topic on nutrigenomics? The term is very new, isn't it? But we have learnt a lot about nutrition and genes. As it is pertinently said, food plays a major role in emancipating disease-free human life. But Hippocrates branded a clear distinction amid food and medicine. To Hippocrates, food is of any edible consumed to offer various bioactive ingredients for the body to maintain a normal mechanism and can be transformed as one of the body components, whereas a medicine is a combination of chemicals that change the body's distorted functional mechanism to normal, but it cannot be converted into body components as like food. Currently, in the field of nutrition, research is underway to find out the relationship of diseases, nutrition, and gene interactions, which is termed as nutritional genetics. Nutrigenomics describes as to how your genes interact with nutrients. It deals with how the genes influence the risk of diet-related diseases and give us a clue as to how to solve the mechanism underlying these genetic predispositions. The main objective of the nutrigenomics field is to establish various metabolic responses towards different types of diet at genetics, that is nutrigenetics, protein, that is proteomics, and metabolite, that is metabolomics, at all these levels in order to reveal the effectiveness of nutrients, which ultimately end up with the development of personalized diet or personalized nutrition based on individual genetic framework. So what do you think are the objectives of this lesson? It is very simple: to introduce the basic concepts of nutrigenomics, to discuss the applications of this field in non-communicable diseases, to offer a new approach for understanding the beneficial effects of dietary compounds on the development of polygenic diseases. In contrast, nutrigenetics involves the study of individual differences of the genetic level that influences individual responses to diet. The individual differences are at the level of single nucleotide polymorphism rather than at gene level. So what do you understand from the term nutrigenomics? Nutrients are the essence of food where the body nourishes on it and sustain a good relation between nutrients and health. The study of interaction between nutrients and body's genome is one such field that is called nutritional genomics or nutrigenomics. It simply portrays the body's genetical adaptation to nutrition interaction. Generally, it is more important to know what sort of gene is responsible for a disease than to know what sort of disease a person has. The underlying mechanism in which the gene responds to specific nutrients need to be examined. The impact of a diet to individual's genetic disposition, its manifestation as single nucleotide polymorphism, that is SNP, and epigenesis are discussed in this particular field. Due to the emerging and growing popularity on nutrigenomics, the need for other advanced omics such as transcriptomics, metabolomics, epigenomics, they are also of high concern in accordance with nutrigenomics, as this is high time that one need to know how the nutrient from diet interact and bring about changes in their phenotype. Based on this, if the nutrient requirements are designed to suit an individual's genetic variations, will probably help to prevent or revamp the disease so that possible health can be accomplished. Both nutrigenomics and nutrigenetics do have altered impacts in two different approaches, either by affecting the genetic mutation or by disturbing the frequency of genetic transformation, but have the same target in bringing out better health through nutrient gene interaction. So this field uses the genetic profiles to classify the patients according to their disease rather than on their symptoms, and the genetic profiles are used to implement targeted therapies aimed to improve or prevent disease progression. So earlier we saw some of the terms as omics. So what are omics? Coming to the first concept of transcriptomics, which is the study of complete set of RNAs, that is the transcriptome, encoded by the genome of a specific cell or organism at a specific time or under a specific set of conditions. What do you mean by metabolomics? It is the field of studying and analyzing molecular complexity of life through the metabolites produced by a cell, tissue, or at organism level. What is the concept of epigenomics? It is the study of epigenetic modification across the entire genome. An epigenome consists of chemical compounds and proteins that can attach to DNA and helps in the production of proteins in particular cells, or it may either control the whole mechanism. What is proteomics? It is the study of the complete structure, function and interactions of the proteins produced by the genes of a particular cell, tissue or an individual organism. So what are the basic concepts in nutrigenomics? The nutrient gene interaction has been based on three chemical classifications, or you can say as three chimerical classifications, which are named as direct, epigenetic and genetic variations. What is direct classification? The nutrients from diet bind with its receptors and act as transcription factor and then interact with the DNA to cause the genetic expression. What is epigenetic? So in this classification, the transcripted nutrients that cause changes in the DNA at its structural level resulted in continuous or persistently altered genetic variation. Coming to the third classification, genetic variation. Genetically altered single nucleotide polymorphism ends up various metabolic changes and directly proportional to changes in the nutrient requirements for any individual. Applications of nutrigenomics. So it has numerous applications. What are they? Coming to the concept of epigenesis. Gene expression may be altered by altering chromosome structure through epigenic or epigenetic mechanisms. Nutrients play an important role in epigenetic mechanism by altering either the DNA methylation or the energy balance of a cell, which is connected to chromatin remodeling enzymes. Deficiencies in choline, methionine, folate, vitamin B12, vitamin B6 and riboflavin of course affects the epigenesis process. Coming to the concept of nutrigenomics and human health, this is very important. Here, the nutrients which plays a decisive role in the progression of disease or well-being of a human being depends upon the individual dietary response. Though the underlying mechanism is still a mystery, numerous factors are considered to be accountable, like their age, gender, body mass, genetical variations and other lifestyle modifications that are related as outcomes. The main purpose of applying the nutritional research over genetical variation leads a promising scope in identifying the precise individuals who can be benefited from arriving particular diet interventional strategies. Almost all genes are merely coded perfectly for all the humans. Some genes do have few changes in the sequencing of amino acids, so they are called polymorphism or single nucleotide polymorphism, in short as SNP, and these are the one which usually shows the genetic differences. These SNP are responsible for the metabolic changes in relation to nutrients which alter the DNA and further persuades the genetic expression. Due to advancement in the field of nutrigenomics and its relation to other high-end omic technologies, it is now easier to study the complex interactions of possible gene and nutrients. What do you mean by genome-wide association studies? So this is in short called GWAS, and let us see the role of GWAS, and it is considered to be a common method which uses the DNA microarray chip technology, which further help to identify the genome using software. After understanding the altered genomic sequencing with the help of nutrigenomics, new functional diet will be developed. The diet will act then on single nucleotide polymorphism. Coming to obesity, it is a very big worldwide problem, isn't it? So let us see what is the relation between obesity and nutrigenomics. It is one of the worldwide problem, as I said earlier, that arise out of lifestyle modification, which pose a serious threat to human population and also the base for other disorders to develop and also for the progression of other lifestyle disorders. Several studies around the world already evidenced the genetic variations and the contributing factors that influence the gene that induces obesity. Though in earlier days obesity was treated in terms of energy balance, the metabolic rate maintenance and prescribing the drug based on individual responses. Now, due to the advancement of the field of nutrigenomics, studies are focused on studying the single nucleotide polymorphism and its DNA fragments related to obesity. The difference in SNP will disclose the individual's susceptibility to obesity development, and tailoring drugs accordingly to individual genetic variation has been enhanced. Similarly, the dietitian also can prescribe diets which are rich in nutrients as well as bioactive compounds which will alter the expression of an individual's genetic makeup. Coming to another disorder, that is known as cardiovascular disease. So next to obesity, CVD has been notably recorded as a chronic non-communicable disease. Progressive evidence has been attempted in indulging the relationship between the development of cardiovascular disease and lifestyle pattern. The latest studies show the influence of genotype on the responsiveness to dietary factors that may reduce cardiovascular disorder risk. Among the several dietary factors, fatty acid, that is endogenous as well as exogenous metabolism, plays a vital role in the development of cardiovascular disease next to gene and hormonal variations. So let us see now the role of PPAR gamma in intestinal fatty acid oxidation. It is well established that PPAR serves as a master regulator of fatty acid catabolism and regulate the following functions. PPAR gamma regulates intestinal cholesterol flux, and it also regulates intestinal nutrient transport and metabolism. PPAR gamma also regulates intestinal motility. PPAR gamma diminishes effects of oxidative stress. So what happens? Therefore, PPAR might be therapeutically valuable for patients with inflammatory bowel disorder or bowel disease. These mechanisms are involved in the control of carbohydrate and lipid metabolism, cell differentiation and growth. Apart from energy yielding property of fat, it also influences the regulation of genetic expression, peroxisome proliferator activated receptor, a type of transcription factors, detect the dietary bioactive component from the diet, further persuade the genetic expression of protein and metabolite formation were considered as dietary signatures. It has been precisely validated and documented that PPAR act as a chief regulator of fatty acid catabolism, which regulates the intestinal cholesterol flux, regulates the intestinal nutrient transportation and metabolism. It also regulates the motility and diminishes the effects of oxidative stress. All these mechanisms are involved in the control of carbohydrate and lipid metabolism and of course cell differentiation and growth. On application of Nutrigenomics, which simply inspects these dietary signatures in specific cells, tissues and organisms level in order to understand the nutrient gene homeostasis. Further, it also helps to recognize the genes that influence the risk of diet-related difficulties on a genome-wide scale and to understand the underlying mechanism of these genetic predispositions in cardiovascular disease development. Coming to the disorder diabetes mellitus, there is no doubt that diabetes mellitus is one of the pandemic disorders that keeps on affecting the world population. Usually, the treatment regimen involves the control of glucose intake and biological uptake mechanism regulated at various levels of targeted organs like pancreas, liver, intestine and other adipose tissue and muscles. The etiology of diabetes mellitus has been widely studied. Certainly, genetic makeup of an inherited individual is also responsible for diabetes mellitus. Single nucleotide polymorphism that influences the phenotypic Changes such as height and weight are also responsible for susceptibility to diabetes mellitus. Nutrigenomics offer a wider scope in treating diabetes mellitus, and several genes have been identified so far which are involved in diabetes mellitus progression. Some of these genes are likely to be regulated by diet, since certain diets are risk factors for diabetes mellitus. Few of the examples are seen in this table, like you can have the dietary factors as, say for example, more carbohydrate intake, which is type 2 diabetes mellitus risk, and magnesium intake also affects fasting glucose level. So what about whole grain intake? Again, this has an implication on fasting insulin level, and omega-3 fatty acid intake lowers the risk of type 2 diabetes mellitus, and of course zinc and magnesium again reduces fasting glucose level, and adherence to Mediterranean diet also is a risk for type 2 diabetes mellitus. And coming to one or two factors like, say for example, trans and cis beta carotene and gamma tocopherol intake affects, or you can say reduces, the risk for type 2 diabetes mellitus. Having an understanding over the genes and nutrients that pose specific threats, Nutrigenomics offers a wider scope in identifying the concerned gene, protein and nutrient interactions. Nevertheless, the technology has become more accessible. It holds complex data pertaining to specific nutrients that hypothetically interact with all other genes in the human genome. Till today, the underlying gene-nutrient interactions for the development of diabetes mellitus are not fully perceived, since the intricate genetic homeostasis and the role of dietary markers in the etiology of the disease and its related traits will probably exist as a mere complexity at the molecular level. The incidence and severity of type 2 diabetes mellitus is significantly associated with the intake of excess calories, dietary fat, high glycemic index carbohydrates and certain micronutrients. Coming to another disorder known as cancer. This is yet again a dominant chronic disease with alarming increase at present and in the future, and this opens up a challenge in treating and controlling the disorder. Extensive data has been published and focused on dietary factors responsible for cancer susceptibility. Yet, irrefutably, there has been unpredictable causative factors which persuade the cancer, such as the exposure time to both the endogenous and exogenous carcinogens, multifarious food pattern, types, and of course preparatory manufacturing practices of food products, genomic dissimilarity, and invariably these factors influence the metabolism. As a result, it's of high importance to acknowledge the role of nutrigenomics to have a collective and comprehensive information about gene polymorphisms involved in cancer growth or development, identifying the target genes that regulate cellular pathways, and improving modern diagnostic tools to control cancer or in terms of prevention strategies. Changes in the carcinogenic metabolism through nutrigenomics is one promising way in which bioactive food components that help to minimize the risk of cancer. The metabolism undergoes two phases, that is phase 1 and phase 2 metabolism. Phase 1 is via cytochrome P450 mediated reactions like reduction, hydrolysis and oxidation, which converts the components into active metabolites. Whereas coming to phase 2 reactions, they are via xenobiotic enzymes and it undergoes various conjugation reactions that turns the active component to inactive and are continuously regulated by different signaling pathways. So let us see how the nutrient and gene associated dietary factors interact in the development of cancer. And this diagram shows the interactions of how the dietary factor influences as well as reduces the risk of cancer. To conclude this lesson on Nutrigenomics, the concepts and applications, we have seen at various levels the basic concepts and the various applications. So what do we end in this conclusion is that the integration of genomic science with nutrition and when possible other lifestyle variables also interact. Nutrition therefore modifies the extent to which different genes are expressed, and more recently the definition of nutrigenomics also encompasses nutritional factors that protect the genome from damage. Hope to see you in the next class.